Good afternoon, everyone. My name is Joshua Kim. I'm a master's student at Louisiana State University. Today, I'm excited to present some results on the differential effects of low salinity and high temperature on diploid and triploid eastern oysters. So let's get into it. All right, so the genetic difference between diploids and triploids is that diploids have 2 sets of chromosomes, while triploids have 3. Now, the primary advantage for triploidy in aquaculture is that triploids show increased growth and yield when compared to diploids while they're grown out in the field. Now, this advantage may come from the fact that triploids show reduced gamete development in compared to diploids. So whereas diploids may be using a lot of energy to make eggs and sperm during the spawning season, triploids may be using this energy directly to just grow bigger and grow faster. So all these factors make triploids a popular option in oyster aquaculture today. All right, despite these advantages, recent studies and observations from growers have shown an elevated mortality of triploids versus diploids, especially during the late spring and summer during low salinity and high temperature conditions. One study is shown in that graph to the right that shows 2 different strains of triploids had a higher cumulative mortality than 3 different strains of diploids at a low salinity site in coastal Louisiana. So these findings are of particular concern to Louisiana and Mississippi due to the high levels of freshwater influence that these states receive. So primarily, this freshwater influence comes from the Mississippi River flow, which dumps around 600,000 cubic feet per second of fresh water directly into the Gulf between these 2 states. On top of that, man-made river diversions in Louisiana release high levels of fresh water directly into our marshes during times of Mississippi River flooding. This fresh water goes from the marshes directly into important oyster growing areas like Breton Sound. So a combination of these kind of result in low and variable salinity in our states, as you can see by the salinity maps shown in the right. On top of this, though, climate change kind of makes this a more relevant problem for everyone, as coastal waters are only expected to get warmer. All right, so how do oysters kind of deal with these changes in salinity? Before we can get into that, let's talk a little bit about osmolality first. So osmolality is a measure of dissolved particles in a solution or in an organism. So in an organism like oysters, osmolality is commonly referring to the amounts of salts and amino acids inside the oyster, whereas osmolality of the seawater is mostly just talking about the amounts of salt. So oysters can adapt to different changes in these salinities by osmoconforming. So organisms have kind of 2 different techniques of coping with changing salinities. The first is this strategy called osmotic regulation, in which an organism maintains a constant blood osmotic pressure despite any changes in the environment. The second strategy, which is what oysters do, is osmotic conformation, in which they change the osmotic pressure within their bodies to match the outside salinity. So therefore, how well oysters can osmoconform can ultimately play a huge role in how well they can survive. So osmoconformation takes a lot of energy for the oysters to perform, and during periods of drastic salinity changes, oysters may even completely shut, which eliminates feeding. So you can imagine maybe if you were an oyster and you had a harder time osmoconforming, you'd be using a lot more energy and be feeding for a less amount of time. Okay, so let's kind of tie this all back up into my lab study. So we saw in the field an elevated triploid versus diploid mortality during that late spring to summer period. These researchers attributed this mortality maybe to low salinity and high temperatures. However, these studies were conducted in the field where there's a variety of different factors at play, such as food availability, differences in water quality. So therefore, we wanted to compare diploid and triploid mortality in low salinity and high temperature conditions in a controlled laboratory study where we can isolate just these 2 factors as direct contributors to the triploid mortality that we see in the field. So throughout this lab study, we'll be tracking mortality daily, and one kind of metric to examine the rate of mortality is this thing called lethal median time, also known as LT50s. So this is just the number of days that it takes for half of the population to die. So on top of this, we'll be comparing the blood osmolality of diploids and triploids in our study, and this could just maybe give us some more insight as to why we saw a difference in mortality. So to do this study, we had to get some oysters first. The oysters that we used were produced at Auburn University Shellfish Laboratory, which were then grown out at Grand Bay Oyster Park until they were around 1 year old. From there, we brought them back to LSU, and we made sure to gather oysters of similar shell height in order to get rid of the effects of size on differential mortality. All right, so here's just a quick snapshot of the experimental design that we did in the lab. So the first kind of period that we had in the lab was an acclimation period. We set the temperature and the salinity kind of similar to the conditions found at Graham Bay at the time of retrieval. And as you can see, we set up 12 different tanks and put in 40 diploids and 40 triploids into each tank. We then increased the temperature in some of our tanks to hit 3 different temperature treatments of 24, 29, and 34 degrees Celsius, while keeping salinity the same in these tanks. This was just to test the effects of high temperature stress only on the differential mortality of diploids and triploids. And so after we held these conditions for around a month, we then decreased the salinity in some of our tanks to get 2 different salinities in each temperature treatment that we established in the previous phase. So this is where we kind of get into that combined stressors phase, where we look at the combined effects of high temperature and low salinity stress on differential diploid and triploid mortality. We have a salinity of 2 and 18 at each of the 3 temperatures that we established earlier. So I also mentioned earlier that we were going to measure osmolality. So we measured osmolality after a 1 to 2 week exposure to the combined stressors phase when mortality was just getting high in some of our tanks. We were a little worried about sample size. So the first step to getting osmolality was to notch the oysters using an angle grinder. We notched these oysters on the dorsal side to expose the abductor muscle. From there, we draw blood using a syringe, and we can separate the blood and plasma by using a centrifuge. We then stored the plasma in the freezer, and once we had all the samples ready, we thawed the plasma and then determined osmolality using an osmometer, which is that device that you see in that bottom picture. All right, so let's kind of get into some results. So here's the results for the high temperature stress phase only, where we had 3 different temperature treatments and kept the salinity the same at 18 parts per thousand. Just to orient you guys to this graph a little bit, we got the cumulative mortality on the y-axis, we got number of days on the x-axis, and we got 3 different graphs just to represent the 3 different temperatures. And we have diploids in blue, triploids in red, and you'll notice a D and a T in the corner of each graph with subsequent number of days. These are the LT50 values, so remember that's the amount of time that it took for half of the oysters to die. And in the parentheses, we got a 95% confidence interval. And in some of the following slides, you won't see a parentheses and instead see an A or a B, and that shows a significant difference. So the first thing you may notice about this is that the cumulative mortality stayed low in all the treatments. None of the diploids or the triploids in any of the temperature treatments exceeded 15%. We also see that the triploids tended to die faster at all the different temperature treatments. When you look at the LT50s, the triploid LT50 values are always lower than the diploids, which just means that they're dying faster. Okay, so here's the results for that combined temperature and low salinity stress phase. This is the results for combined stressors at 24 degrees Celsius. We have 2 different replicates, and we have the salinity of 18 in the top kind of row, and then salinity of 2 on the bottom row. So we see here that the triploids are dying faster than the diploids at 2 parts per thousand. When you look at the LT50s on the bottom row, you'll notice that the triploid LT50 is around half that of the diploids in both replicates. And we see that at 18 parts per thousand, the LT50s are somewhat similar. So for replicate 1, they're pretty similar. And in replicate 2, we saw a significant difference, but the difference isn't nearly as big as at a salinity of 2, and the end cumulative mortality is pretty similar. So here's the results for the combined stressors at 29 degrees Celsius. We see here again that triploids are dying faster than diploids at 2 parts per thousand. When you look at those LT50s, the triploid values are around half that of the diploids in both replicates. At 18 parts per thousand, this time we see kind of a variable type of mortality, where in replicate 1 we saw a significant difference, but in replicate 2 the LT50s are nearly identical. All right, so here's the combined stressors results at 34 degrees Celsius. We see in this phase that the triploids are dying faster than the diploids at 2 parts per thousand. So once again, we see that the triploid values are around half that of the diploids. And we also see here a 100% cumulative mortality in less than 25 days. This was the fastest mortality treatment that we had out of all of them. And then we also see that at 18, we see some pretty interesting results where the diploids actually died faster than the triploids at 18 parts per thousand in both replicates. This was the only treatment in which we saw this type of result, where the diploid LT50s are lower than the triploids. Here's the results for the plasma osmolality. So just to orient you guys to this graph, once again, we got osmolality on the y-axis. We got the 2 different salinity treatments, 2 and 18 on the x-axis. Diploids are shown in the blue boxes and triploids are shown in the red boxes. And you'll kind of notice 2 dotted lines in each of the graphs. So that's the expected osmolality at 18 parts per thousand, which is that top one. And then for the bottom dotted line, that's the expected osmolality at a salinity of 2. So whichever box is kind of closer to that dotted line means that's the box that's osmoconforming better. So we noticed that at 18 parts per thousand, the osmolality for diploids and triploids are similar across treatments and even within treatments. You'll notice an overlap of the boxes in every single treatment, and the boxes are all in the same place across the different temperatures. However, after a change of salinity from 18 to 2, we notice that the triploids are not osmoconforming as well as the diploids. This kind of trend is most evident at 24 degrees Celsius. As you can see, the blue box is way closer to that dotted line than the triploid is, and at 24 degrees, there's no overlap at all, which means that the diploids are significantly better at osmoconforming. Okay, so our main finding here was that the faster triploid mortality that we saw was happening at low salinities, especially at higher temperatures. This could be due to the fact that the triploids were not osmoconforming as well as the diploids at this extreme low salinity, which confirms recent studies done by Cassis et al., which found that triploids were slower and less efficient in conducting biochemical processes within their cells. The lack of osmoconformation might be explained by the larger cell size of triploids. So triploids in previous studies have been shown to be around 10% bigger in their cell size than the diploids, and these larger cells have lower surface to volume ratios. So if you look at that kind of just diagram that I made on the bottom, you'll see that for a diploid with an example radius of around 10, the surface area to volume ratio shown in red is 0.3. But when you increase that radius by around 10% for a triploid cell, you'll notice that the surface area to volume ratio drops 10% to 0.27. So having a lower surface to volume ratio ultimately means that you're going to have slower transport processes across membranes that are critical to cell regulation and balance. Okay, so we saw also that the triploids were surviving better than diploids at the higher temperatures. This could be due to the fact that the triploids have a higher body energy reserve. So we just mentioned that triploids have around a 10% bigger cell, and within these bigger cells, they may have more reserves to deal with single stressors, like high temperatures. However, under combined and multiple stressors, we see that the cellular energy demand is greater than the cellular energy mitochondrial output in triploids versus diploids, which could result in a higher triploid mortality, which is what we saw in our study. Okay, so our main bottom line here, our take-home message, is that triploids are certainly more sensitive to low salinity at higher temperatures than diploids are. When we kind of tie this back up into the grand scheme of things, we see that we originally did the study to further investigate that differential mortality that we saw in the field. We noticed that these triploids were dying faster than diploids, especially at lower salinities and higher temperatures. However, we know that there's other factors to be investigated. There's other groups of things that are contributing to this differential mortality. So these other groups may include other environmental factors such as food availability, differences in water quality, also questions about reproduction. How much more energy are these diploids investing into reproduction than triploids, and how does that affect the mortality? Different husbandry techniques that growers may be using. So what kind of techniques, such as the stocking density, such as desiccation, these types of things could have an effect on that mortality. And finally, we got the all-encompassing kind of question of genetics. So who these oysters, where they came from, who these parents are, is there any sort of inbreeding? These are all topics that can certainly contribute to the differential triploid versus diploid mortality that we see in the field. All right, with that, I just want to say a huge thank you to our co-authors and to our funding agencies, and I'd be happy to take any questions. Thank you, guys.
All right. Do we have any questions for Joshua? Got one back here.
So, what do you?
Sorry, is someone else talking? Oh, okay. Thank you. I was wondering, are you guys planning to do some transcriptome analysis of those oysters, triploid, diploid, because I think that may provide some insight into exactly what's going on, especially in response to low salinity, for example.
Yeah, I'm sorry. I just kind of missed that question. What type of analyses were you asking about?
Transcriptomic mRNA.
I'm certainly not planning on doing those types of analysis, but I know that members of my lab are involved in those types of things. So those will certainly be topics that will be further investigated later.
All right, Andy, you get the last question here before lunch. Oh, sorry. Go ahead, Tony.
what do you suspect would be the mortality at high salinity and high temperature? Because I'm asking about, say, the lower Laguna Madre, which is considered hypersaline at times.
Right. The first thing that pops up into my head may be diseases in the water. The first guess is diseases. I know that the higher salinities and higher temperatures are more prone to carrying more types of diseases. But it also could, especially in the Laguna Madre, it could just be that single stressor of higher temperatures. I could see that contributing to mortality for sure.
All right, we'll wrap up with Andy.
Very good presentation. I think your conclusion about the larger cells makes a lot of sense to me. But I've killed a few oysters at low salinity in Mobile Bay in my day, and it's always the biggest ones that seem to have the highest mortality. And I know you tried to get size controlled, but if you have the same size diploid and triploid, you have usually a higher meat or body mass in the triploid, which seems like there's more stuff to keep alive. And what happens in our situation goes down, I think they just quit pumping and they just stay closed and they just burn up their energy sooner because they're keeping more tissues alive. Did you look at the body mass, the meat weights between the two?
We did look at the meat weights. Those are still in analyses, but yeah, we got the meat weights, we got the ash-free dry meat weights, and we are in process of analyzing the condition index results. So I can get back to you on that for sure.
All right, another round of applause for Joshua.